Hi everyone, good morning, good afternoon, and good evening to all the attendees from different time zones of USA, Europe, and Asia Pacific regions. Thank you so much for your interest in Saraca webinars and registering in large numbers for our live webinars. This webinar is on biological evaluation of medical device including changes with 993. Your presence is highly appreciated. I hope you all are doing great and staying. Now, let me introduce myself first. I am Ankit Shukla, Business Development Manager, Saraca Solutions. And yes, I am your host for today's webinar. To our panelists now. So, our lead presenter for today's webinar is Samuel Wade, and I will let you know about him uh, in the start. Before that, I will just touch upon few important points related to this webinar. All participants will be muted during the live webinar session. You can note down your questions during the live webinar and ask them after the webinar ends. Or you can also send us emails, putting questions on it. Our team will certificate of participation to all the attendees who has joined our live webinar. We will be happy to share a recorded version of this webinar at the end to all our webinar attendees. You can also find a recorded version of this webinar and our other webinars on our website's webinar page, which is www.saracasolutions.com. Now, coming to the presenters. So we have two panelists for today's webinar. The lead presenter is Samuel Wade. Sam has a bachelor's degree in science on microbiology and immunology. He has been involved in medical device quality and regulatory for more than 40 years. And he was doing quality and regulatory consultant work and work for ISO 14971, ISO 13485, 510K submissions, and obviously 10993. Now I will introduce our second panelist, who is Ankur Agrawal. Ankur has more than 15 years of experience in medical device industry. He is AVP of Medical Device and Pharma in Saraca Solutions. He will share his thoughts on why compliance is so important for medical device industry. Now I will request Sam to join us. And before we start the webinar, I would like to say sorry to our all attendees because of some technical issues. Sam's camera is, so we are really sorry for that. Over to you, Sam.
Okay, I'd like to thank Ankit for that great presentation, and thank you for giving me such a high regard. Thank you all for coming. I really appreciate everybody being here today on a Friday in the midst of a pandemic. On the other hand, we're all behind screens. You can't see me, and that's really a good thing. So I'd like to move on. I'm going to give you a high-level view of the 10993-1 2018 changes. I'm not going to get into details of specific testing, although I will in some of the questions, and how important this change has been. So the agenda for today is changes in the 10993 section, the application of 10993 to medical device evaluation, risk management as applied to 10993. Risk management is now throughout many of the FDA guidance documents, the 13485 and the MDR. Risk management weaves through all of these documents, and you can't just apply risk management in isolation any longer. And I'll cover briefly the time duration for evaluation of testing. There is a difference between 10993 an FDA 10993 guidance document. And I wanted to point that out because when you go to Annex A and see the table that's in 10993 ISO is quite different from the FDA. The FDA retains the original uh, testing categories with an X at the top of each uh, type of test and uh, with each type of contact. That's different from what FDA requests. So if you don't look at 1093 from the FDA's guidance standpoint, you could run into trouble with your submission in the US. You won't have any issues getting into the NDR using 10993-1 2018. Okay, some of the specific changes in 10993-1. It was approved by, it was actually released by ISO in 2018. It was approved here by Amy in April 24th, 2020. It was approved by ANSI in April 27th, 2020. And it was, well, one of the other things that I should mention about ANSI and Amy acceptance is since that time in 2020, there have been a series of other parts within 10993 that have also changed. And you need to look at all of these parts and see how they're applicable to your medical device with the changes and when they go into effect as you evaluate prior to initiating any testing your device. And those parts include 10993-9, -10, -15, -18, -19, and there's a couple others that are also have been either added or changed in the time frame since 2020. And the last one that was changed was the addition of nanoparticles. So nanoparticles now need to be examined from a risk standpoint, how it's applicable to your device, and whether you need to do additional testing that's recommended, additional searches for use of your material in the scientific literature, or additional non-validated testing that needs to be cleared. The quickness of this change, you can judge by the FDA consensus standard, whose number for ISO and ANSI is standard 2.258. If you look, it was accepted by FDA as a consensus standard in January 14th, 2019. That's a very quick change for FDA to accept an ISO standard. And I just want to point out again that except for table 8.1 annex, that conflicts with the FDA guidance document, and that portion is not part of the consensus standard. You need to go back and look, and I do have to point out it's a little simpler than the one for the ISO standard itself. One of the major categories that have changed is when you look at Annex A1 table, you will notice that they revised the annex with endpoints. Instead of the normal X that is the top of the column, it's now been changed to address endpoints for physical and or chemical information and material-mediated pyrogenicity, chronic toxicity, carcinogenicity, reproductive/developmental toxicity, and degradation. All these have to be evaluated from a threshold standpoint. You cannot contain without adequate explanation and research going beyond those thresholds for each one of those parts and each one of those testing programs. Again, this is it's changed that it now indicates endpoints are to be considered with an E instead of tests to be conducted with an X. The importance of this means that you really need to do your research on your components and your device prior to doing any number one risk management decisions, determining the risk, and actually doing some physical chemical testing prior to doing your in vivo testing. The Annex B was replaced and is now titled Guidance on the Conduct of Biological Evaluation Within a Risk Management Process. And that's formally ISO TR 15499. It is now referenced as an ISO standard within the normative references. That means ISO 14971 is now part of 10993-1. And additional definitions for terms used throughout the 10993 series of standards have now been codified and made the same throughout the series. And there's an additional reference to ISO 18562, all parts, for biocompatibility evaluation of breathing gas pathways and healthcare applications. This has really come into effect during the pandemic with the gas pathway for ventilators, respirators, oxygen concentrators, to make sure that all parts are biocompatible. There's no particulates being pushed into the lungs during the ventilation process. Additional information on the evaluation of nanomaterial in a sorbable material left within the body is now included in ISO 10993-1. Sorbable material means materials left behind after the device has been released into the body. That would be things like sorbable stents, sorbable material for gel foam for sutures, things like that that are evaluated separately and now have additional information on those materials. There's new information about the next two subjects, the evaluation of non-contacting medical devices and transitory contacting the medical device. Those are now better defined and new information is included within the standard. We'll cover a little bit of that later on. There have been significant editing changes throughout the document. By that I mean the King's English is used throughout the document as opposed to American English. The metric system is used, and instead of using dots, they use commas for separation of thousands and millions and so forth. The rationale for change has been an effort both by the FDA and the working group. And this change is based upon not using in vivo systems or animal models as much, unless you absolutely have to. They want to transition to cell lines to prevent pain, anguish, and distress to the laboratory animals being used. In some countries, you can't use animals at all. In the United States and some other countries, you can, but they want you to consider wherever possible in vitro testing, which means cell lines, L929, so forth. The FDA also has a working group on using standard methods, which you should become aware of. This means instead of using pigs and pigs' veins, they're looking at alternate mechanisms for standardizing tests in an in vitro model for example, using standardized models for veins and arteries, if they can be as consistent and mimic the use of the device within the body. It's a good group to get involved with, and these standards will be coming along as they perfect them. The next point that I would like to ensure that everybody understands is this: ensure personnel making the biocompatibility assessment are qualified by training and experience. Prior to this implementation, anybody that worked in the laboratory was considered, or had experience, was considered adequate training. Now they want you to be thoroughly trained by, thoroughly documented as being trained, and have the experience to make biocompatibility decisions for a device. This is a very important one, and you'll see it carried through the remainder of the standard as well. And that becomes a problem with a couple of the parts on dealing with toxicology. I have to say there aren't that many toxicologists around to share between the notified bodies who are going to come in and look at your biocompatibility testing, or FDA, and looking at your biocompatibility testing and assessing whether your personnel are adequate and trained adequately to do the job when there's not enough people to do both sides of it, notified bodies and the companies. I expect that would be rectified over time, but during a recent seminar I went to, it was a major fear that there weren't enough people to answer this change. The next point is, again, something that's carried through all of the 10993, a lot of the QMS systems documents, is to ensure risk associated with legacy, new, or changes in devices use state-of-the-art techniques. That means, and I'm sure that everybody on this call has heard the term state-of-the-art. That means if a test becomes state-of-the-art between the time of the release of this standard to the time you're doing your biocompatibility, you have to evaluate your device under the state-of-the-art techniques. That means you need to go and do literature searches to determine if there have been changes in the way you evaluate materials or devices. You have to do a literature search for additional material that may be out there on the behavior of your material in a biological system, whether you need to check the mod or systems in the in Europe to determine whether there has been any significant events that have occurred with this particular material in the field. And lastly, you need to make sure that your post-market surveillance is working in a good manner to catch any changes that may occur after introduction of the device into the marketing, In another seminar I give on risk management is sometimes done lastly as part of the development process. The new risk management techniques that are being called for need to be done during the beginning of the R&D process. If you have get to the end of your development process and you're getting ready to release into the market, and all of a sudden you have a material, for example, the reds are famous for this, that are toxic, you're going to have to go back and redo your device. Or if you determine that the risk is unacceptable by the time you get ready to release the market, you may have to go back and redesign or change some of the materials in your device, or justify why you did it that way. The traditional way of doing risk management needs to be done right up front during the determination of essential performance of the device in actual use. You can't do it in a vacuum. You need to go out, talk to the people that are going to use this device, learn how they're going to use it, whether they're going to use it in a manner which if you break the device during its use in a manner that you're not familiar with or didn't anticipate, and you expose an interior material that was not tested for biocompatibility, you need to go back and do that. And it may affect your product in the field. It may affect your product in process, and you may need to go back and redo it. So this calls for a change in the way we do to the development process and consider the risk as they're used, the essential performance of the device, and the intended use. That's another term that's being used throughout the standard. You need to consider the intended use of the device when you're doing biocompatibility. Again, that means going out and making sure that your intended use is what it's actually going to be used for and not some whim of somebody that really hasn't gone out and decided this would be a good thing to use in the field. You really got to get to know the users, how it's used, where it's used, the environment, the conditions, and the training of the people using it. This also means that you need to consider what kind of physical and chemical testing or other kinds of testing needs to be done for your devices prior to doing your biocompatibility. You need to go through that first prior to implementation of any in vivo testing or any in vitro testing. This is an initial point I'd like to emphasize again. I can't say this enough, is assuring the standard applies the same risk management approach and philosophies that the FDA and NDR are now requiring. There is a general feeling that the risk management has not done well because we're still having recalls. And recalls we've had a series of recalls this month, which are phenomenal. There are things that should have been considered, batteries dying, connections becoming loose, and so forth. That says that these risks were not either considered during the development process or the essential performance of the device was not adequately tested. I'm going to cover the scope of the 10993 standard, the general principles governing the biological evaluation of medical devices within a risk management process, the general categorization of medical devices based on the nature and duration of the contact with the body. It's a very similar scope to what we've seen in the past. The evaluation of all existing relevant data from all sources, though, is somewhat different for the scope of this standard than previous ones. All we really had to do before was go to a chart, pull out the ones that make sense for the device, whether it's temporary, tissue contact, and so forth, and do whatever the box indicates. This is different. You need to evaluate from all sources relevant data regarding the material you're going to use in the device and the biocompatibility of that material. If you have a legacy device, you need to look at the gaps in your available data set based upon a risk analysis. Doesn't mean you have to go back and redo all your testing, but it does mean you have to take a serious look at any gaps in your testing and whether it reaches the standard where a risk assessment determines that the risk is too high and you may need to go back and redo some of the testing. the identification of additional data sets necessary to analyze the biological safety of the medical device. That's another thing that I covered briefly, but we'll get a little bit deeper into that. The assessment of the biological safety of the medical device applies to the evaluation of the materials and medical devices that are expected to have a direct or indirect contact with the patient's body during the intended use. In other words, the essential performance of the device during its intended use in the field by appropriately trained personnel. The user's body, if the medical device is intended for protection. This is a question that's come up even during the audits by notified bodies. One auditor was trying to tell me that the laser skins, the skin or the outside of a laser, which doesn't come in contact with the patient during use, needed to be tested for biocompatibility. But that's only if you're going to wear the laser as a mask, as indicated here. And this is a direct quote from the standard. And this means if the medical device is going to be worn as a face mask, gloves, operating gown, covers, anything that protects the operator that comes in correct that's intended for protection of the operator needs to be biocompatible. But if it's not used for protection, it does not. Risk such as changes to the medical device over time as part of an overall biological safety assessment. That means if the essential performance of your device in its intended use is said to be in your literature 20 years, then you need to know how that device, implants, for example, changes over time. Does any of the material leach out? If it leaches out, does it pose a problem to surrounding tissues over time at the end of your life? Well, I shouldn't say it that way for implants, but at the end of the device's life, which in some cases can be 20, 30 years. And if a device breaks in a manner that exposes a material that hasn't been assessed for biocompatibility, it now will need to be, particularly which exposes body tissue to new or novel materials. That means you really have to know during your risk assessment how your device is going to perform in the field. Is it going to break in a manner that you didn't anticipate during its intended use? And if it does, are any new materials exposed to the tissues of the body? You need to know that, or that could set your design project back quite a few months and cost you quite a bit of money.
Sam, this is a very important point you are bringing out here. Does it mean all the materials that can potentially be exposed to the body need to be assessed for the biocompatibility?
Only if your risk assessment indicates to you that there's a possibility of breakage, which will expose a new material to the body tissues. And again, that's something that should be done during your risk assessment phase. It should be documented in your essential performance of the device. We all know that doesn't happen all the time, but if you suspect that it may, it would be worth your while to do biocompatibility testing. Thank you for the question. That was a good one. The next thing that I want to bring out are the normative references that are either new or are part of 10993. As you may or may not know, 10993-1 is not just a one-off standard. There are many other normative references that are contained within that pertain to your biological evaluation of your device. I'll start going through those, and some of these have changed. Part 3, test for genotoxicity, carcinogenicity, and reproductive toxicity, depending upon the length of time its intended use and where it's used, may be necessary to adequately test your device. Part 4, selection of test for interactions with blood. Part 5, test for in vitro cytotoxicity. Of all the tests you can run to get an idea of biocompatibility, this is the quickest and cheapest one. You can normally get a cytotoxicity test using L29 and its various formats, MEM overlay and so forth. This will get within a week, and it's cheap. It's within a couple $100, $300. Will give you an idea of whether your material will pass a biocompatibility test. So if you're thinking about using material, this is a good test to use to determine, can kind of give you a feel for whether your material is going to pass or not. I had a material once that I was evaluating. It was made with a red dye, and it killed everything. I didn't even get through the testing period. It just wiped everything. So I never use red anymore in a device that comes in contact with a tissue, because I know it's, at least in my experience, it's been toxic. I don't know if there are now reds out there that could do this, but that's just a little from my background. Part 6 is a test for local effects after implantation. I think it goes without saying that some of the things that are implanted may have things that leach out, and you need to know how these leachates or leachable materials affect local tissues. And you need to know whether they affect them positively or negatively. And if they affect them negatively, you need to be able to determine what kind of risk is associated with that. Part 7, ethylene oxide sterilization residuals. This is pretty much standard, hasn't changed in a long time, although I understand it's under review. Part 9, framework for identification and quantification for potential degradation products. Again, this goes along with long-term devices that are left in the body, or maybe in there for a short period of time, or 30 days or so. You need to know what's going to happen to that device. Is it going to degrade? This particular part 9 and part 10, both of these have changed and have changed somewhat in a dramatic fashion since 2018. So these 9 and 10 need to be reviewed and be familiar with them if it's applicable to your device. Part 10, test for irritation and skin sensitization have dramatic changes in the way they're done. 11 hasn't changed, which is test for systemic toxicity. 12 is sample preparation and reference materials. 13 is identification and quantification of degradation products from polymeric medical devices. Part 14, identification and quantification and degradation products from ceramics. Hasn't changed, that goes back to 2006. Part 15, identification and quantification of degradation products from metals and alloys. This has changed. And some of this is applicable to cobalt as part of your steel being used in implants. 16 is toxicokinetic study design for degradation products in leachables. 17 is establishment of allowable limits for leachable substances. 18 is chemical characterization and materials. 19 is physical, chemical, morphological, and topographical characterization of materials. These two, when you start getting into how this reads, you really do need to have somebody who is trained as either a chemist or a cytotoxicist, excuse me, a toxicological trained person, because some of these tests and whether you need to do all of them or not is dependent upon their decisions and their assessments. Part 20, principles and methods for immunotoxicity testing of medical devices. And lastly, 23, a new one, biological evaluation of medical devices, test for irritation. Lastly, you'll notice that a normal reference, which I've outlined here in blue, and I kept going, it didn't. Sorry about that. Is the inclusion of ISO 14971, 2007 medical devices, application and risk management to medical devices. As you can see, 2007 is the one that's included, but there is a newer one that's out that comes into effect shortly. So you need to look at your risk management system as it's changed and it's applicable to this 10993 material. I'm going to cover a couple definitions that I consider important that people have asked about and what they mean. Biocompatibility is defined in section 3.1 as the ability of a medical device or material to perform with an appropriate host response in a specific application. Again, this goes back to how is your device going to perform in its intended use and whether that will be biocompatible or not. Biological risk is a combination of probability of harm to health occurring as a result of adverse reactions associated with medical device or material interactions and the severity of that harm. Biological safety is freedom from unacceptable biological risk in the context of intended use. And again, intended use, essential performance, you got to go back and look at how that's being used. A chemical constituent is defined as any synthetic or natural substance that is used in a process for manufacturing materials and or medical devices, including the base materials, additive, antioxidants, UV stabilizers, color additives, dyes, etc., and processing aids, which may be used during the milling process or the machining process, anti-foaming agents. All those have to be determined upfront to determine whether those are biocompatible in the context of its intended use. I know that when you're extruding material, there are a lot of additives in extrusion for stabilizing material, preventing it from turning yellow during ethylene oxide radiation. And you got to know not only that it's in there, but if it's in there in the concentration that goes over the threshold, or if it leaches out, how does it affect the human body? The final product is a medical device or medical device component that has been subjected to all manufacturing processes for the to be marketed medical device, including packaging and, if applicable, sterilization. Sterilization can affect your packaging materials. For example, if you have craft paper with ink, that ink may leach into your product during the sterilization process. So you have to be aware of that. Material characterization is a broad and general process of collecting existing information about a material's chemistry, structure, and other properties, and if appropriate, new data to facilitate the evaluation of these properties. And finally, nanomaterial, material with any external dimension in the nanoscale or having internal structure or surface structure in the nanoscale. Physical and chemical information. knowledge regarding formulation, manufacturing process, geometric and physical properties, and the type of body contact and clinical use that is used to determine whether additional biological or material characterization testing is needed. Risk analysis comes right out of 495, systemic use of available information to identify hazards and to eliminate this risk. Toxicological risk is probability of having a specified degree of an adverse reaction occurring in response to a specified level of exposure. A toxicological risk is a limit, such as a tolerable intake, tolerable exposure, allowable limit, or threshold of toxicological concern, below which adverse effects are not expected for relevant biological endpoints. Risk management, systemic application of management policies, procedures, and practices to the task of analyzing, evaluating, controlling, and monitoring risk. Risk evaluation is a process of comparing the estimated risk against a given risk criteria to determine the acceptability of the risk, and risk assessment is the overall process. General principles is the biological evaluation of any material, medical device intended for use in humans shall form part of a structured biological evaluation plan within a risk management process. The risk management plan should identify aspects of biological evaluation requiring specific technical competencies and shall identify the persons responsible for the biological evaluation, and it should contain their CV in there as a method of demonstrating competence in the area. General principles, a general safe history of use and specified role and physical form that's equivalent to that of the medical device under design. That is, if the testing, if the material is in use, has been used for many years, and there's enough data out in the field which you can document demonstrates the biocompatibility of a material, they may not have to do any additional testing. And that's where your risk assessment comes in right at the very beginning. And in some cases, that would save people a lot of time. And it also helps in the selection of materials to be used in the medical device manufacturer. The first considerations for fitness of purpose about characteristic properties of the material, which include chemical, toxicological evaluation, can include both a review of relevant existing preclinical and clinical data and actual testing. Such evaluation may result in the conclusion that no testing is needed if the material has physical, electrical, morphological, and mechanical properties that are consistent with biocompatibility.
Sam, in case of equivalent device, do manufacturers need to produce a rationale or any report of the existing equivalent device?
Yes, you do. You need to document whatever your rationale is for why you're using that device, and not only the rationale, but the documentation to show that you've considered all the parameters, the research in the field, examples of the material in the field, and so forth, to demonstrate that you've done the job as showing that it is biocompatible. A good question. Description of medical device chemical constituents and consideration of material characteristics shall precede any biological testing. Chemical characterization with an appropriate toxicological threshold can be used to determine if further testing is needed. And that's where those two, 17 and 18, come in that have changed in the past 2 or 3 years. The physical effects of the medical device shall be considered if they can impact the biocompatibility. All known biological hazards shall be considered for every material and final product, but this does not imply that testing for all possible hazards will be necessary or practical. Test results can't guarantee freedom from potential biological hazards. Thus, biological investigation shall be followed by careful observation for unexpected adverse reactions or events during clinical use of the medical device. Again, that's what they're going to expect from a biocompatibility standpoint. Are you seeing any adverse events coming in through your post-market surveillance that indicates a new biocompatibility issue is there? And if it is, you have to run through your risk management process and determine whether that risk is acceptable. In vitro tests, which are appropriately validated, reasonably and practically available, reliable, and reproducible, shall be considered for use in preference to in vivo use. Whenever in vivo tests are indicated by findings of the initial risk assessment, use of the appropriate in vitro screening, if available, should be considered before in vivo tests are commenced. In certain circumstances, such as specific medical devices or biological endpoint assessments, if a non-standardized, non-validated test is necessary, additional information regarding the rationale for the study design and data interpretation should be provided. The biological safety of a medical device shall be evaluated by the manufacturer over the whole life cycle of a medical device.
So is there any certain frequency defined in the standard at which the biocompatibility assessment has to be done after product launch?
There is no frequency specified. You, as part of your risk assessment in your design, need to consider at what point would be appropriate to do testing for degradation of your product over time. I don't think it would be appropriate to do time 0 and time 30 years. So you need to pick out something that you can defend and that would make sense from literature search and study the materials in use in a biological system. Protection of humans is a primary goal of this document. A secondary goal is to ensure animal welfare and to minimize the number and exposure of test animals. Pain and suffering or distress to the animals be minimized. Testing will be performed on the final medical device or a representative sample from the final device or materials processed in the same manner as the final medical device. And testing will be performed in the same manner as the intended use, taking in consideration essential performance. Degradation of the product in vivo should be considered when any part of the device will remain within the body. The mechanisms of the degradation process, including rate and extent, shall be determined. For example, if you have a stent that's implanted into an artery that degrades over time, you need to know how that's going to perform from a biological performance standpoint. If you have a stent that's metal, what is the effect on the body fluid on that stent as it remains within that artery or vein for the length of time and the essential performance of that device? That concludes my portion of the presentation. I'd like to turn it back over to Ankit for any questions that may arise. I want to thank you for attending. It's a lot of material to cover, and I only covered it at a 20,000 foot level. I really recommend that you purchase or find a copy of 10993-1 and read it thoroughly and become familiar with it. It will be part of your future as time moves on, and it's going to be more and more difficult as time goes on. I don't think we're stopped done with these type of evaluations, and that we'll see future standards come out in the future or significant modifications being made. Thank you for your participation. Ankit?
Yeah, thank you, Sam. So, hi everyone. Now you can just put your questions in the chat box, or you can raise your hand if you have questions from Sam. I will unmute you and you can ask your question. Or if you are not comfortable on the audio, you can also put your question in the chat box. I will uh, giving your answers. Okay. Question, Sam, from you. I'm just forwarding it to you. Sam, can you see a question here on your screen?
Actually, I can't.
She is asking, could you give some examples of which test would you perform in these two devices? Screwdriver, metal, no contact to the patient, and screwdriver, metal or plastic with long implantation time.
Well, for the non-contact screwdriver, you'd have to determine what it's made out of. And I would rationalize that one on based upon what you've learned from the field on it and not do any testing from what I understand. I don't hold me to that, but I don't, there's enough material out there of these type of products being used in the literature that I think you probably could justify writing that up to, by somebody with adequate training and experience, and that would be your conclusion, that no further testing is needed. The implantable, I would have to know the materials, uh, how long it's implanted for. I would have to know a lot of stuff before I could make a recommendation. Be happy to talk to you about it, but it'd be hard for me to make any kind of opinion without knowing more.
Okay. So we have another question from James. I will read it for you. Do you have any suggestions for using existing materials to stabilize biocompatibility of a medical device, external only?
Could you read the question one more time?
Do you have any suggestions for using existing materials to stabilize biocompatible device?
You're asking for a recommendation for material to be used in a device to stabilize it. Again, it's hard for me to analyze something and give you a rational or my opinion upon what you can be used without knowing what its essential performance is, what the material is that it's going into. That's something that I would recommend that you do research again, like I told the previous person, and make a determination as to what you need to do. The Annex B, I believe it is, can give you some guidance in that context for you to walk through that. Again, I really sincerely think and hope that you will read 10993-1 from start to finish. It's not that long, but there's a wealth of information about how you go about doing exactly these kind of problems in the annex, in the text of the material, excuse me, the text of the standard. And these will answer a lot of your questions and give you guidance as to how you go about doing some of this stuff. And again, you're going to need somebody that has a background and the experience of making these kind of decisions documented.
Sam, we have one more question from Anika. She is asking, where should a person being for a device? There would be nothing substantial to find in the literature.
So it's a new material. If there's nothing in, well, if there's nothing in the literature and it's a brand new material, you're going to have to do extensive testing or a rationale as to why you're not going to do it. I would recommend, again, you talk to a toxicologist about this material and do, again, your risk assessment, look at the intended use, what the essential performance is expected of this device, and then go to what kind of testing would we need to do to prove that to somebody. That's the best I can do for you.
One more question is there, Sam. When we do material change and the new material has the same CAS number with the existing material, need to do the biocompatibility test? If yes, what is the minimum test required for that test?
A CIS number doesn't necessarily mean it's the same material. It just means that it's similar. There could be other materials in there that if you've ever seen chemicals delivered to a site, they come in railroad cars and things that are added to them. I've actually seen a person get up there with a 5-gallon pail of additive, take his calibrated hand, reach into the 5-gallon container and throw it into this 30,000-gallon railroad car. Now, you know that one guy's hand, even though it may be calibrated, is going to be different than another's. So there's going to be slight differences in chemicals. What you need to do is compare those from a rational basis, determine whether there is enough material out published on those materials. There's plenty of, when it comes to a chemical, there are plenty of characterizations made and analysis done that are public. And you could go through those and you can either come up with a recommendation that we're not going to do any because it's exactly the same, or we are going to do a cytotoxicity just to make sure. So again, there's lots of things that you can do. And that decision, that if you do come up with that, it needs to be documented like you would any change in anything, that we did a literature search, we did this, these are the people that were involved in that decision, we consult with so-and-so and decided that we don't need to progress, and make sure that it makes scientific sense and it's rational and it's from based upon state of the art, and you should be okay.
Okay. So Maria is asking one more question, Sam.
Sure.
Give a small explanation about the, sorry, explanation about the ID class 3 implantable device.
He's talking about class 3 from a use perspective or class 3 from an FDA perspective. Either way, if it's an implantable from a class 3 perspective, either in the EU or FDA, substantial amount of work is going to have to be done if it's an implantable. You're going to need to know the long-term consequences of implanting that into the human body. You're going to need to know whether it degrades, how fast it degrades, what are the degradation byproducts. all of those kind of things with any class 3, if it's an implantable, will need to be thoroughly investigated. Or again, if the rationale is that it's been done before and you're using a material that's well classified and well documented, you may not need to do that. And again, I would refer to some materials that are used for implants. Have been around for an awful long time. Their degradation rates and everything are quite well known. And again, if you can rationalize and have enough material out there in the public domain or even private domain, doing your own internal testing and preclinical and clinical results, you can use that. So again, it depends upon your specific device. It's very difficult for a consultant to come up with the kind of answers you're looking for without knowing a lot more about your product and its use.
Maria, you have more questions. I think you can ask your questions on audio. I'm just giving you, yeah, you can ask it.
Okay, thank you. Good morning, good afternoon, and good evening to everyone. Well, indeed, my question, well, I just using class 3 device. I already wrote it, class in long-term implantable joint replacement class 3 device, just to put some kind of quite critical device. It was just to have something in mind. But indeed, my question was about the testing. Which testing? Which are the goals about the testing? I know a lot of names, so the GCMS, the ICP, the TOC, the particle test, Biobander, LLA, and cytotoxicity. I just wanted to know what are the goals, because always when I ask my company to make some tests or something, all the people ask me, what do you want to know? If you know the question, you can make a test and you get the answer. So indeed, what is the question we have to ask us to ask a laboratory to do some of these tests? I mean, ICP, TOC, particle test, Biobander. And also if I can ask you already the second question, also what are the good tests to do also cleaning and cleaning validation?
On cleaning validation, there actually is a guidance document you could use as a basis for that. FDA, and I believe Europe also has a guidance document on validation of various cleaning methods. Some of the ones at FDA get quite detailed depending upon the instrument, like an endoscope has reprocessing and validation cleaning documented. For the specific kinds of tests you would need for a device, you go to Annex A, and if your device is a long-term implantable, now a class 3 device, critical device, doesn't always mean it's implantable. For example, if you're making a heart-lung machine or a blood pump, it's a critical device, but it's not implantable. You still need to go to the 10993 Annex A and look at how long that's in contact with. For example, that's blood contact direct. And a surgical procedure may take, I don't know, if you're doing implanting an LVAD, for example, maybe an hour. If you're doing a complete heart replacement, that may be 5 hours. So your device would be in contact with the blood from anywhere, for worst cases, what you got to plan for let's say 5 hours circulating within your device. So you go to the annex and you look at blood circulation long term and do the appropriate test that's called out for in the standard. And that's the basis when somebody asks you that question, well, why do we need to do the test? Well, it's because that's what the standard calls for. If you want to sell in the market, in Europe or in the FDA, in the United States or Canada or a few other countries, you need to do the testing to meet the standard. And that's all spelled out in 10993-1. And that's why I highly recommend you read it. It's very valuable to read. It's like you hate to read 13485 because it's so long and involved and detailed, but in order to understand the 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 standard, you really do need to read it and understand it. And a lot of answers to your questions are in there. But it's a lot of reading, I have to give you that. You know, if I have trouble getting to sleep, I try to read those just before I go to bed.
Okay, so Sam, we have many questions over there, but we are already getting late. So we can we extend this for 10 more minutes so that we can pick questions over there and then we can close after 10 minutes.
It's okay with me.
Okay, so Preeti is asking if any magnet is shielded with a implantable instrument, shall that be considered for testing?
If the could you say that again?
If any magnet is shielded within a non-implantable instrument. If the magnet is a shield within a non-implantable instrument, shall that be considered for testing?
Well, what I would do is I would document whether it should be or shouldn't be. Again, I would go back. The chances of the encapsulated magnet coming in contact with the body is pretty minimal. So I would suggest you consider the possibilities of that happening. Let's say that the magnet is encased with plastic and the plastic breaks and the magnet pops out and becomes in contact with the patient or falls into an open surgical area. Then I would consider doing testing on it. If it's firmly in something and the chances of it breaking free and the chances of it coming in contact with the patient are minimal, I would write it up that way and not do any testing on it. But again, you have to consider whether we will or not document why you wouldn't do the test. And that's easy. Again, it's 5 or 6 minutes and maybe a half an hour of your time to document that and put it as part of your plan.
So our next question is, Sam, is this biocompatible study carried out by the manufacturer or by the end user? What is the role for end user? This question is from Joby.
Well, biocompatibility testing is done by the manufacturer. It's not done by the end user. The end user wants to get a product, open it up and take it out of the package and use it. So the emphasis, if you're going to introduce a product into the marketplace anywhere, it's up to you to determine whether that device is biocompatible, and that's your responsibility. And it's very unlikely you would get approval for that device without having some kind of data to prove that one way or the other.
The next question is from Anu. She is asking what section would be applicable to infusion systems? Considering the patient, it is 10993 part B, and for operator, what would be the test?
For the operator that's using the infusion pump, there isn't any because he's not coming in contact with the device. I'm assuming that I make a couple assumptions here that the infusion pump uses closed circuit tubing, uh, like an infusion set. And then you attach that to the infusion pump. I don't even know if the infusion pump needs to be biocompatible depending upon the design, but the infusion set would definitely have to be. And the infusion set would need to be biocompatible for a fluid pathway for the most part, unless you're pumping blood. But they call it fluid pathways, definitely need to be tested for biocompatibility, leachables and that sort of thing. Again, it goes back to how your device is used, the design of your device, the intended use, and what the critical essential performances are for that device. Where would they be used? How would they be used? And who's going to use them?
Okay, so I'm going to ask the last question. You can still put questions in the chat box. We will be really happy to answer those questions over your email IDs. So the last question for today's webinar is for ISO 10993-7, what is the difference between neonates and pediatric?
Go back and look at what a 7 is. Um, 7 applies to ethylene oxide residuals. So you're talking about ethylene oxide itself, ethylene glycol. So the question is, what is the difference between neonates and what was the other one? I got the impression that the question dealt with the age of the patient. Is that true?
Yeah, it may be.
Well, the standard for ethylene oxide sterilization residuals calls out that you cannot have more than a certain milligram percent or milligrams per whatever for each of those residuals. That hasn't changed in a long time. So the way to rate that is if you go over that, you can't use it, period.
Sam, can you go back to our presentation?
Well, I'm looking at 7 is part 7, which is what was asked, is ethylene oxide sterilization residuals.
Okay.
And what that means is ethylene oxide, when you use it as a sterilant, leaves it behind certain residuals or the breakdown of the ethylene oxide in contact with water and so forth. And one of those is ethylene glycol. There are a couple others, ethylene chlorhydrin, and those all have values that you can't go above. And if you do, you can't use them. Now, there are a lot of ways to deal with those values. For example, most companies, when it comes out of the sterilizer, will put it through a couple of heat cycles to drive the ethylene oxide out of the packaging. And you can vary the length of those heat cycles. Some people will just stick them in a warehouse for 7 days and let it blow, and then you test it at the end of the 7 days. Or you could test it at the end of 2 weeks until your residuals get down to a level that are not, or that are in line with the standard. That's one way of dealing with that. It's not very often that a residual will stay in there forever. So you can usually get rid of it or get it down to an acceptable level by just letting it stand in a heated room or whatever your SOPs call out for.
Okay.
Okay.
Sam, I would request if you can go back to the original presentation so that I can connect with the peoples.
Oh, I'm sorry. This one here, right?
Yeah. Then 21 slide, can you go on? The next one. Yeah, you can click.
Do what?
Your slideshow is, yeah. One more click, please. So this is a very important announcement for all the attendees. Our next webinar will be on in medical device manufacturing. How important is that? And please mark your calendar for 21st July 2021, same time as this webinar was there. And I put few poll questions here for your feedback so that we can take more quality webinars to you. So I'm going to start sharing polls here. That will be really helpful for us to give you more information on topics and webinars, and we can improve our quality for you as well. Okay, so our next poll is here.
Do I hit that little thing there?
Yep. And the next poll is, this one is very important. We can do a webinar as per your choice of topic. So you can just let us know which topic would you prefer for our next webinar. Last poll is here. Sam, could you please move to the next slide? Okay, so a lot of attendees were asking about will they get a recorded version of this webinar and a copy of this presentation. So yes, you will get it. And in addition to that, you will also get a certification for this participation in our live webinar. And questions, we have tried to give answers of all your questions, but we have a lot of questions over chat box. Really happy to answer those questions over your email IDs. So my contact details are on the next slide. If you have more questions and you need more information, you can just contact me or my email ID, which is ankit.shukla@saraca.com. You can also contact Sam for more information. We will be really happy to answer your questions and queries. So thank you for joining our live web. It was really successful and huge registrations were there. We are really sorry because of attendees limit. Some people were not able to join. So next time we will plan something big where we can have more people in our live session. Thank you. Have a great day.